Hello, biology class, and today we are going to start a new unit on molecular genetics. And in order to understand molecular genetics, we need to go back and look at the process of mitosis, a type of cellular reproduction, because we're going to introduce another cellular reproduction process called meiosis. And you probably already heard of that, but we're going to look at it very in depth. And in order to do that, let's review our mitosis first. So if you all remember, we have our cell cycle right here, exemplified. You remember this from, I think it was Unit 5 or something. The cell cycle is simply the sequence of growth, metabolism, and division for the cell. Right here, we have all of it. The whole thing is called the cell cycle, right? Interphase is just this period starting here and it goes around to here. It is most of the cell's activities happen during interphase. The cells go about doing their normal job. We do have DNA replication right here in part of the interphase, but the majority of time is spent in interphase where the cell just goes about its normal business, whatever it normally does. We have this little part of the cell cycle right here called the mitotic phase, and that includes mitosis and cytokinesis. Now remember from the cell cycle, mitosis is where we have the equal division of nuclear information. The chromosomes are sorted out so that we go from one cell to two new cells, two daughter cells there. Cytokinesis is just the final division of the cytoplasm and the organelles. And that makes up the mitotic phase. So let's do a quick review about chromosomes. Tie all this together. We've just been talking about chromosomes. When we look at a cell, any cell in an organism's body, we can look at the nucleus here. We're talking about eukaryotic organisms. Look at the nucleus, and we have our chromosomes located in the nucleus. That's where we find them. And if we pull one out right here, we can see it a little bit better. You can see that located on that chromosome is a gene, many genes actually. Quite a few genes are located on the chromosome. Here's a better picture of that. Here's a gene and another gene. It's a segment of DNA that codes for a trait. Here, up close, we have our structure of our chromosome. We have in the middle the centromere. It holds the two sister chromatids together. Remember, those two sister chromatids are identical copies. It was copied in the S phase of interphase. Remember that we get one, this pairing, we get one chromosome pair from mom and one from dad. We just learned about that when we were talking about alleles and genetic traits that are passed on. Also something to note, the number of chromosomes varies a great deal in different species. It can be anywhere from 6 to over 200 different chromosomes. When we talk about human chromosomes right here, we have 46 of the diploid chromosomes. This is diploid. It's the duplicated chromosome with the two sister chromatids. When we look at a fruit fly, it only has 8. A pea only has 14. But just so you aren't wondering if your intelligence has anything to do with the number of chromosomes, you have a potato has more than us, 48, and a goldfish has 94. So that's just a little interesting side note that different organisms all have different numbers of chromosomes. Okay, let's go back and do a very short, quick review of mitosis. Let's start with interphase. When we have a cell that is in interphase, it's going about its normal cell activities. Remember, we have our G1 where it's normal cell activities. The S phase during interphase is where the chromatin is duplicated. The DNA is all duplicated. We get that those two sister chromatids, right? G2, we go back to normal cell activity. Organelles are also duplicated at this time, some of the organelles. The chromosomes are not visible during interphase. So that is something, an important fact to remember, that we don't see any of the chromosomes. They are they're in here, but they don't appear vivid under a microscope because they haven't condensed. So when that cell enters the mitotic phase, and it starts mitosis. Remember, we have our little mnemonic, P, M, A, T, and cytokinesis. PMAT. Remember PMAT? P for prophase. It begins the mitosis. The chromosomes become visible here, and the nuclear envelope starts to dissolve around it. The centrioles migrate to opposite ends, and spindle fibers start to grow so that we can set up for metaphase. Metaphase is where the chromosomes line up along the equator. The spindle fibers in the centrioles, they attach to the centromeres of each of the chromosomes, and they pull them into a line. There's a checkpoint for accuracy here as well. Anaphase is our next phase, where we are pulling the centromeres are being split apart in the chromosomes, and the spindle fibers shorten to pull those chromosomes away from each other. And you can think anaphase, they are going away, and they are being pulled to opposite poles. The cell elongates somewhat. Telophase is where we have two now sort of groupings of chromosomes. It's the reverse of prophase. The chromosomes relax back into the chromatin form, so you start to not be able to see them as well. The nuclear envelope reforms around each of them. And then the very final step in all of this, where we have cytokinesis, the cytoplasm itself separates and the organelles separate. Remember, in plants, we get a cell plate that forms. In animals, the membrane just pinches in, we get a cleavage furrow, and we have two new daughter cells. So, more review. What is the result of mitosis? When a cell goes through this mitosis process, the result is two identical daughter cells. Those daughter cells have the same number and the same kinds of chromosomes as in the original cell. They're able to then go through their interphase. Each of the daughter cells, when they go through the S phase, they will double their chromosomes again. Their chromosome replication, they will get double the DNA, so they will have the sister chromatids, and they can go through this whole process again and make even more cells. But the result is two identical daughter cells. Let's talk just a little bit here about single-celled organisms versus multicellular organisms. Single-celled organisms, paramecium, things that are just one cell big, they never have more than one cell, they actually reproduce via mitosis. Binary fission, they go through binary fission, and in order to get a new organism, they go through this process. Multicellular organisms, however, do not reproduce via mitosis, right? Mitosis for multicellular organisms is very important to replace or repair different cells. When your skin cells die, you need new skin cells, so your body goes through mitosis to make more cells. When the organism is growing, when you're a baby, you certainly have fewer cells than you do as an adult, and you need a lot of mitosis going on in order to grow. When your tissues are differentiating, you're getting new tissues, and they're, for example, your heart is a different type of tissue than your skin, and mitosis is pivotal in forming the different organs and the differentiated tissues in your body. So, if multicellular organisms are not able to reproduce via mitosis, how do they reproduce? Multicellular organisms are involved in a process called meiosis. This is the specialized cell division that results in gamete formation. What are gametes? And I think we've hinted on this a little bit, haven't formally introduced it. Gametes are the sperm and the egg. The sex cells are gametes. And how do we form gametes? They're very, very different than all the other cells in your body. They have half the number of chromosomes as all the other cells in your body. And that's so that when they combine during fertilization, you don't have way too many chromosomes. That is a process called meiosis, and we're going to look at that right now. So let's go back. Mitosis is the regular cell division. It gives us two identical daughter cells, right? PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. These daughter cells are diploid. They have pairs of chromosomes. When we form these cells, they're able to go through the interphase and double their genetic material. We have diploid cells here. They have the full number of chromosomes. If we had two regular diploid cells that combined, okay, they would form a cell with way too many chromosomes. This would not work for gamete production. If we look at N as the number of pairs or different kinds of chromosomes, the human cells in your whole body, except for your gametes, your whole body, all the cells contain 2N, or 46 chromosomes. Those are diploid. That's the diploid number of chromosomes for all the cells in your body except for your gametes. So if our eggs and sperm were also diploid, then when they fertilized, when the egg and the sperm fertilized, the next generation would have 92 chromosomes. It would be what we call tetraploid, or 4N. It would not be viable. It wouldn't even be able to function. It wouldn't make any sense. In a very short number of generations, if it were to work and the organism were to live, cells would be just absolutely overwhelmed with chromosomes. They would just keep, every generation would have double the number of chromosomes. Well, how do we solve this problem? Meiosis solves this beautifully. Let's look at that. Meiosis creates the gametes, the eggs and the sperm, with only one set of chromosomes, half of what the diploid cells have. We call those haploid, okay? The egg and the sperm each have half the number of chromosomes as the rest of your body does, and we call those haploid. Just an abbreviation, haploid is N. Remember, diploid was 2N. That's the rest of the cells in your body, okay? So when we have these haploid gametes, the egg and the sperm are haploid, we get those. We have the haploid with 23, the haploid sperm with 23. We get they combine during fertilization. Fertilization creates a diploid cell called a zygote. That zygote is just the fertilized egg. It's diploid. So let's look at how this occurs. How does meiosis accomplish this tremendous feat to have the number of chromosomes so that we can have fertilization and maintain a consistent number of chromosomes in the organism's body? Here's how we do it. It's very similar to mitosis in some ways. We still have PMAT. PMAT is still in effect. However, there are two rounds of division. We have Meiosis I right here, and then we have a second round, Meiosis II. In the first division, in the first round of cell division, Meiosis I, the chromosome number is halved. It's cut in half, and we get two cells with half the number of chromosomes. Then in the second division, we have sister chromatids that are separating, and they are, each of those in turn are producing two more daughter cells, and we get, in the end, 4 daughter cells. There's some very key differences, and we'll talk about that in a little bit, but let's just go through these first two processes, the first 2 rounds of division in meiosis, and look at them a little bit more in detail. Meiosis I, here's Meiosis I. We start with our regular cell in interphase, just like with mitosis. We're starting in interphase, but when this cell undergoes Meiosis I, we have prophase, and we call it prophase 1 because it's in the first round. We'll have metaphase 1, anaphase 1, and then telophase 1 in cytokinesis. This is all Meiosis I, the first round of division. In prophase, this is very different from mitosis. Each of the chromosome pairs with a corresponding homologous chromosome, a chromosome that is the same in length, okay? So these are homologous chromosomes. They're the same length, and they pair up together, and here are two homologous and two homologous, and they form what's called a tetrad, okay? Tetrad is a really important key term for meiosis. Tetrads do not happen in mitosis. And it is then the tetrads that line up along the equator in meiosis for metaphase 1. Okay? Then in anaphase, when the chromosomes are split up and pulled apart away from each other, the tetrads separate. Not the individual chromosomes, but the tetrads separate. Okay? Then here we are at telophase 1 in cytokinesis. We have the nuclear membrane reforming, very similar here to the mitosis, and we get the cells separating into two daughter cells. Now we're ready to enter Meiosis II, the second round of this meiosis process. We do PMAT again, but it will be prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay? And this process is very much like mitosis. We enter prophase, where these two daughter cells, we'll just take one of the daughter cells, for example, from our meiosis one. We'll look at this daughter cell, and the nuclear envelope sort of dissolves. We get the chromosomes that come and then line up along the equator for the metaphase 2. Then they are pulled away or apart in anaphase 2, and they reform the nuclear envelope starts to reform during telophase 2, and then cytokinesis separates so that we end up with two daughter cells for this one and two daughter cells for this one. So ultimately, total, we have 4 daughter cells. Let's look at a comparison now between mitosis and meiosis. Obviously, some similarities. They're really evident. Interphase, the same, right? There's DNA replication in the interphase time that's needed for both the meiosis and mitosis. The general order of PMAT events is the same in both. Cytokinesis is the same in both. So what's different? Let's highlight some of these differences. In mitosis, here's mitosis. During metaphase, the chromosomes line up, single file, right? And whereas in meiosis, in metaphase 1, we have the tetrads lining up. So the chromosomes are lining up double file in formation because they're in the form of tetrads. Another difference would be the results. In mitosis, we get 2 diploid cells, and those daughter cells are identical to the parent. Big, big, big difference with meiosis. Our results are 4 daughter cells. They are unique. They are haploid. They are gametes. So very different. They're not diploid, they're haploid, and they are all unique. None of them are like their parents. Some other differences here. We have in meiosis, in prophase 1, just the fact that there are tetrads. Obviously, mitosis doesn't have tetrads. And very key important thing in meiosis, there are 2 cycles of PMAT instead of 1 cycle. So now that you know why meiosis is so important, come to class, explain that to me, explain the process of it, differences between meiosis and mitosis. Here's some advanced ideas. Ask your own questions. Why can only one sperm fertilize an egg? Why are women over 40 more likely to have chromosomal problems in their children? Down syndrome. How does meiosis explain why horses and donkeys can successfully interbreed, but their offspring are sterile? What is polyploidy? How does it relate to meiosis? What's the effect of it on plants? It's kind of a fun word to say. So I look forward to seeing you in class and seeing what you have to show me.